Choi, Head of Drug Discovery at Verge Genomics, presenting Accelerating Discovery and Development of ALS Therapeutics with an AI-powered all-in-human platform. Welcome, Irene.
Hello, thank you very much. Let me get my window situated. I apologize for the, this was the best I could do for the slide format. My PowerPoint and keynotes were not having it with Zoom today. So hopefully you can see the slide. Please feel free to stop and interject. I do realize I'm the block between you and the break. And so I'm happy to just kind of go through this and focus on kind of the points of interest that pique questions. But thank you very much for this opportunity to present. We are very excited to be able to share the work that we have done at Verge Genomics to really enable progression of our ALS therapeutic program, kind of leveraging the AI machine learning approach. So today I will highlight how we take advantage of AI-enabled machine learning to not only identify a disease signature and novel targets, but also then in turn use that to accelerate the discovery and evaluation in our preclinical models. And then lastly, focus a little bit into some of our methods in validating translational biomarkers that has really prompted us to be able to get into the clinic, hopefully in a few weeks. Great. So as many of you know, kind of looking back in oncology and the progress that's been made in developing effective therapeutics, you know, it's really, the field has advanced really through the application of human genetics. And if we look at the failures in the past several decades for neurodegenerative disease, we're now seeing a trend in finding meaningful therapeutics. The most notable here is Spinraza, which is the first disease-modifying therapy approved for treating spinal muscular atrophy. And prior to this, there really wasn't any known therapy available for these children that have neurodegeneration resulting in paralysis. And after the approval of Spinraza, it really paved the way for validating the use of leveraging human genetics, identifying targets that are derived from those gene mutations or associations that really drive the disease phenotype, and really expanding the way into possibilities for other more meaningful therapeutics to be developed. As you can see, you know, after Spinraza, others were able to quickly come up with FDA successes in additional therapeutics to treat SMA. And so we are kind of now at this point where it's accepted that using this targeted genetic approach is really the key to unlocking success in neuroscience and treating neurodegenerative disease. And companies now see a viable path forward in making meaningful therapeutics. So we first begin by integrating human genetics with tissue analysis to define functional pathways through known genetic mutations of disease, or so-called genetic drivers. Our platform begins with identifying from human genetics and genomics, well-validated genetic mutations that are known to cause neurodegeneration. So I'm going to use Parkinson's here as an example, but later in the talk, I will be speaking to our ALS program. So here's an example where we have a known genetic mutation associated with Parkinson's disease, in this case, a synuclein. Then we use large proprietary data sets of human tissue RNA sequences to map disease-specific gene networks associated with those genetic drivers and the functional consequences that lead to disease. And while most efforts have really been focused on primarily suppressing the directly observable phenotypes from the mutation itself, we seek instead to understand the function of the gene itself. And with this, our platform identifies druggable and differentiated targets that functionally counteract the genetic shortfalls and not specific to a given mutation. And this allows us to first use that genetically defined population as a proof of concept to de-risk our clinical approach. And because we focus on convergent pathways that are shared by multiple genetic drivers to identify targeted therapeutics, this provides us with a natural expansion and to potential multiple patient populations. So using this approach, I developed a portfolio of programs that functionally counteract well-validated disease-causing genetic deficits. We have our first program in ALS, which focuses on a genetic mutation that results in a loss of function for C9orf72 that in turn causes ALS through a loss of lysosomal function and activity. And so our lead program, which I'll go into a little bit, describes how we're able to not only identify that deficit, but restore it with our therapeutic. More importantly, what we were able to do with our disease signature identified from our network analysis is that this dysregulation is also not exclusive to C9 patients, but also found in sporadic patients, which really allows us to be able to expand our therapeutic potential, not to just a specific genetic population, but to the broader sporadic community. We also have additional programs in our Parkinson's pipeline, one that is around PINK1 mutation, with showing activity in mitochondrial dysfunction. And we have another program in Parkinson's kind of related to synuclein mutations and lysosome dysfunction in biology. So a little bit into our ALS disease signature, ALS disease network, and some of kind of what we were able to identify by leveraging AI-enabled machine learning algorithms. And so this is a representation of our ALS disease network. It's composed of about 300 genes. And through comprehensive analysis of public and proprietary data sets, our platform has generated a gene co-expression network that is significantly and differently expressed in ALS patient samples versus healthy controls. And this gene signature actually demonstrates convergent pathways that are shared by multiple genetic drivers that are well known to be causal in ALS and neurodegenerative disease. And in fact, this gene network that we identified is consistently downregulated across multiple studies. And this network is actually 5.4-fold enriched for ALS Mendelian genes, as well as 9 molecular processes that have previously been implicated in ALS. So this gave us a great degree of confidence that this gene network that was initially identified from sporadic patients could actually be meaningful to the development of ALS disease. And so taking a look a little bit further with the original network that we identified, we were able to show that C9orf72 was located as one of the central hubs in the network, giving us more confidence in the relevance of these network of genes. This network is also enriched in lysosomal function and activity, which many of the literature have now shown the role that lysosome function and activity and dysfunction plays in a disease progression for ALS. And moreover, we were able to show that with repeated studies, I think across 1,000 postmortem brains and spinal cords, this network is consistently and significantly downregulated, giving us more degree of confidence that this is a differentiator between ALS patients and healthy individuals. So a little bit into kind of the C9orf biology. I probably don't need to explain too much. So, but feel free to stop and ask any questions. As many of you know, as the field has really focused, the work has really advanced and progressed into understanding the toxicity associated with the gain of function mutations, here using C9orf72, an example that results in this toxic protein aggregates, the DPRs, that result in neurodegeneration and motor neuron death. And with our collaborators in 2018, we were able to show that there is actually haploinsufficiency that results in impaired lysosomal function with a loss of function activity of the C9orf72 mutation. So it's not just a gain of function protein aggregates, but also a loss of function in lysosomal activity that it kind of contributes to the disease pathology. And further work has been done with this collaboration to show that restoration of this lysosomal dysfunction can actually not only improve the clearance of DPR, but also enhance the glutamate receptor turnover. And this is really what we feel sets us apart versus what has been done in the field in terms of the treatment approaches for targeting the aggregates. By restoring the lysosomal dysfunction, we're able to not only improve the clearance of autophagy, but also improve the receptor turnover that contributes to glutamate excitotoxicity. And in this publication, with our partners at USC, we really explore that therapeutic potential of one of our targets. So we looked at PIK5 kinase as a target that was central in our network. And by inhibiting this kinase activity, we were able to show that the presence of dipeptide repeats in these patient-derived neurons are actually reduced dramatically in culture. Moreover, we were able to measure survival of these neurons in a dish to show that we can restore the patient-derived neurons to those of healthy individuals when we treat them with our PIK5 kinase inhibitor. Moreover, we were able to show that PIK5 is efficacious in an in vivo model where we see a reduction in the glutamate receptor puncta, and this is in the C9 mouse. And in the knockout mouse, we're able to show reduction in the infarct and necrosis due to the excitotoxicity with an injury model of NMDA, showing that there is proof of concept here in PIK5 kinase inhibition and the therapeutic potential as it relates to the toxic aggregates and the loss of function activity. And so I'm going to go now into our program. So after this collaboration, what we have done is created a whole library of PIK5 kinase inhibitors. These are new chemical entities that we improved upon what was commercially available and known out there to really hone in on not only the potency and activity, but also the oral bioavailability. As you can imagine, patient compliance and therapeutics are more accessible if they can be just taken at home on some sort of a daily frequency. And so we really focused on this as a central approach into making a therapeutic that could be more broadly accessible to patients. And in our small molecule program, what I'm showing here are just a series of curves where we've been able to show improvements over the different iterations of our small molecules for oral bioavailability in the mouse. And moreover, we were able to demonstrate that PIK5 kinase inhibition potency significantly correlates with disease rescue using an in vivo model of HEK cells that are transfected to express a TDP43 mutation, show an accelerated death, but with PIK5 kinase inhibition, we're able to see a nice relationship with potency and activity to show these cells survive longer. Now, going back to what we do from here, one of the challenges in drug discovery is the use of the relevant preclinical models that we can then use to be able to identify and predict what would be a meaningful therapeutic that we can advance forward. And so we lean back on our initial disease signature, the ALS disease signature that composed of about 300 genes that's downregulated in ALS patients. What this shows here is if you take this network of genes and then kind of line them up in a row, so each bar represents a gene. Blue indicates that it's downregulated with respect to healthy individuals. What we're able to show is that in sporadic patients, this network is downregulated. That's how we originally identified it. with C9orf72 being central to the network, it's actually more downregulated. It's a stronger downregulation identified in patients with C9orf72 mutation. Moreover, we're able to use the same network to look at preclinical models to see which of these models could better mimic what we're seeing in patients. We actually also used the TDP43 mouse model, the RNLS8, that was described in the previous talks. And we were able to show that this network is also consistently regulated and recapitulated in this mouse model, giving us confidence that this would be the relevant model to test. We also tested our in vitro model system, looking at iPSC motor neurons derived from SOD1 patients, C9orf72 and TDP43, and we are able to show that it recapitulates the same downregulation in this ALS disease signature. And so we use those model systems to then identify potential efficacy of our small molecule therapeutic. And what I show here is basically those Kaplan-Meier curves, but simplified in a bar to show the extent of rescue with the treatment of our drug. So on the left, it's those iPSC motor neurons. This is using SOD1 as an example. So a ALS patient with a SOD1 mutation, the iPSC-derived motor neurons show a deficit in survival compared to a healthy control or isogenic control. We're able to then look at the survival or improvement in survival in these different patient-derived iPSC motor neurons with the treatment of our small molecule. And here's just an example of one of our earlier generations of PIK5 kinase inhibitor and a later generation where we're able to get a near restoration to healthy levels and more so in a second generation compound that we developed, showing consistent rescue that is seen across genetic mutations. And so the next question is, how does this really compare to the other compounds that have been tested or treated or approved for use? And looking at the same model system and testing the previously approved drugs, riluzole and edaravone, as well as late phase 3 candidates that weren't unable to show a clinical benefit, what we're able to show that in our model system, we really don't see an improvement in survival of these motor neurons, really speaking to the fact that our small molecule could have a real significant therapeutic benefit into improving the longevity of the motor neurons that are affected in disease. Then we further characterize our small molecule by looking at compound activity, bridging between in vitro and in vivo model systems. We quickly validate target engagement using an immediate downstream cytokine of PIK5 kinase activity. It actually is involved in the production of IL-12P40, which is downstream cytokine. And what we show here is that both in the in vitro setting as well as the in vivo, we get this really nice concentration dependent change or inhibition of IL-12P40, showing significant target engagement, both in a human cell model system, but also in an intact organism using the mouse, to verify that our drug is getting in, it's orally available, and it's acting in the way that we anticipate it to without significant loss in potency or activity. Then we expand on that into looking at really improving the gap in how we identify that our compound is hitting the target, as well as is it engaging the pathway that our hypothesis suggests that is being needs to restore. And then lastly, is our compound behaving in a way where we can have early indications of drug efficacy in treating patients? And looking kind of across those three lenses into our translational biomarker program, we first demonstrate, which is what I showed before, an in vivo application of target engagement. Is it hitting the immediate downstream activity? Do we see changes in the relevant cytokine? And in fact, we see a really nice dose-dependent reduction in IL-12P40. On the left is the vehicle control, and we use just a strong anti-inflammatory as our positive control to show the extent of reduction of reducing the pro-inflammatory cytokine. Then we look at and measure an additional biomarker associated with lysosomal dysfunction. And this is actually done in the TDP43 mouse, where we show that in the context of disease, we see this significant elevation in this lysosome-associated biomarker. And with increasing doses of our drug, we actually are able to get a significant reduction. And this was measured in brain. Further, we want to be able to connect that relationship to what is being measured to predict disease progression in patients. And with a neurofilament light really standing out as a way to track and measure disease progression, we did just that in the same mouse model, where in the diseased mouse that's untreated, you see this significant elevation, and this is measuring neurofilament light in CSF. And you can see that with our highest dose, we get this nice significant reduction of neurofilament light. This is a very aggressive model. I think some of the other speakers have also emphasized this. The animals succumb to their disease within a matter of 4 weeks. We have tested other approved drugs, other therapeutics. We haven't seen any other small molecule or therapy really make an impact on neurofilament light in this model. It's super aggressive. But we have been able to demonstrate now with several of our compounds that PIK5 kinase inhibition actually does show an improvement. And then going back to that original disease signature, are we able to then restore the network of genes that we have originally identified to be dysregulated with disease? And, the punchline is down here where, in those same animal models where we see a rescue, not only in the lysosome-associated biomarker, but also neurofilament light, we also see a restoration of that same network of genes that were downregulated in disease. And this gives us a lot of promise that the therapeutic is actually working in a meaningful way that can modify and mitigate disease progression. And so in conclusion, I wanted to update here. We're very excited that we have a development candidate. It's a potent PIK5 kinase inhibitor that's orally available. We've been able to demonstrate strong oral availability as well as brain penetration across rodent and non-rodent species. We have demonstrated rescue in ALS disease models, both in vitro and in vivo, much like the examples that I showed you before. We have been able to demonstrate our development candidate to have robust PK-PD relationship with a target engagement biomarker, as well as demonstrating significant rescue in neurofilament light in the CSF. We actually also have measured some behavioral phenotype in that model and have also shown some delays in clasping behavior, which is a sign of progressive degeneration in that model. And more importantly, we have completed our GLP tox studies and demonstrated that this compound is safe. And we filed our register for the clinical trials, and our phase 1 study is set to start in a few weeks. We expect our first in human to be just a few weeks away. And so we are very excited to be now at a point where we can advance our therapeutic and really hope to make a meaningful difference in really pushing the boundaries of bringing new therapeutics for treating neurodegenerative disease. And that's it. Thank you very much.
Thank you for an incredibly comprehensive presentation, and congratulations on the progress moving toward the clinic. A question from Dr. Benjamin Brooks, and it relates to you showed gene downregulated pathways. Do you have a parallel program that reveals pathways that are upregulated?
Yeah, that's a great question. In our gene network analysis, we look at the whole genome, and our computational biologists really take a look at the different networks of genes that are differentially regulated. And some do go up, some do go down. Some of them have opposing relationships to each other. We are also able to identify networks of genes that are more compensatory to the disease rather than causal to the disease. So for instance, in our ALS program, there were networks of genes that were more associated with the inflammation and being upregulated in the context of disease. And so we really look at all the different groups of genes or modules, as we call them, to see how they are with relation not only to the causal aspects of disease, but also in response to disease. One of the tricky parts, though, is the gene expression network that we get is from postmortem tissue. And so it's a little hard to then extrapolate a time-dependent change where some of these network changes where it's compensatory would really make an impact. And so for the moment, we've focused on looking at the causal network, but we are looking more broadly across our platform to see what other types of information we can glean from, especially if we're thinking about biomarkers and how we track disease progression or translate that into other biofluids. Um, and some of our targets do require an activation or overexpression instead of an inhibition, where we were able to pursue here.
Thank you. I've been undisciplined as a moderator with everybody's time, and
Yeah.
we're well past our time.